Hey, everybody, welcome back to the I Believe podcast. I'm so glad you guys could join us today. And we're bringing back one of our most popular guests from the beginning of when I started doing the podcast. This is Dr. Justin Moser. A lot of you guys have showed up in his clinic and said, this is Danae's doctor, right? So we are so glad that he's willing to be a guest on the podcast and just for his expertise and his continued research in the field of ocular melanoma. But he's a medical oncologist at the Honor Health Research Institute in Arizona. And today we're going to be chatting about the INSIGHT Biobank. So I don't really know a whole lot about this. So Justin, do you want to give us a little bit of a rundown? Like, what is the biobank and why do we feel like it's super important right now?
Yeah, absolutely. So first of all, thanks for the introduction and thanks for having me. So a biobank to start is really an area or it's a process where we collect biospecimens. So that could be samples from patients. So that could be blood, tissue, vitreal fluid from the eye, really anything. And the goal of a biobank is to collect tissue from enough patients that we can collectively look and understand the disease. These are really important for rare tumors like uveal melanoma because really to understand how a cancer works, you typically need a lot of samples, sometimes, hundreds, sometimes if not thousands of samples. And when you have a disease that's as rare as uveal melanoma, we need to get the number of samples needed to truly understand it and therefore truly treat it. We need to collectively get samples as a big group because no one institution can ever collect enough samples themselves.
No, I feel like that totally makes sense. And I feel like this is something that even from the time that I was originally diagnosed almost 6 years ago, that I've heard kind of noise in the community about like, oh, like we need to bank tissue. We need to be able to research like, and I think I had like the castle biopsy was the decision next for uveal melanoma. And I remember asking or like learning about how like that sample, that little teeny tiny little like needle biopsy that they do, that they store an extra bit of that sample. And I was like, oh, well, like what can we do with that? The biobank sounds like that would be something, that it could be used in, or, not necessarily that sample specifically, but just, when there are extra samples from a biopsy, whether it's from the eye or somewhere else, that would be helpful. So like, where was kind of the brainchild and like the vision for creating the INSIGHT Biobank specifically?
Yeah, I think the cool, unique thing about the INSIGHT Biobank is it's really driven by a Cure Insight. It was really a patient-driven thing. And a couple of years ago, Cure Insight led an effort of, trying to get a bunch of institutions together to create a biobank. And one of the barriers we had, which is one of the barriers we have with all institutional biobanks, is getting, if institutions are involved, then it's hard to get agreements on how the tissue can be used, who owns the tissue, who makes the deciding factors. And so a Cure Insight, really led by Melody, the president, spent a long time trying to figure out how to get around this. And she's really the brainchild and the person who got this up and running, where the INSIGHT Biobank is really a patient-driven biobank. So instead of institutions coming to you and saying, you as a patient saying, you're going to get a procedure, can we take extra tissue? It's really a Cure Insight messaging to the patients and the patients going to their providers saying, I want to contribute extra tissue to this. And then, kits are sent to the patient or to their surgeon, to their interventional radiologist, and it goes to the patient-run biobank. To make sure that it is patient-run, it is governed by a group of individuals, including a couple of medical oncologists like myself, a couple of ocular oncologists, we have pathologists, we have patient advocates, and we have people from a Cure Insight to make sure that we're allowing the samples to be used to push the science forward, but also in a responsible way. If patients are going to donate tissue and donate their time to contribute to the biobank, we want to make sure, one, that we can actually get the tissue out to people who want it and who have a good idea, but we're also only using that tissue for stuff that's going to move the field forward.
Yeah, that makes sense. Like we want to make sure it's used judiciously.
Exactly.
um, So, all right. So, a biobank, like you said, it exists to help kind of try to answer some of these research questions in fields that are more rare. Uveal melanoma, we know, has been notoriously difficult to get like large scale movement forward. And obviously, in the last 5 or so years, we've seen some really good jumps in research, in clinical trials. But what research questions have still kind of continued to be difficult or hard to answer without a resource like this biobank?
Yeah, a lot of it comes down to just what is the biology of uveal melanoma, right? We know that 96% or so of uveal melanomas are driven by mutations in GNAQ or GNA11. But what that actually changes and what downstream proteins and signaling, that actually are subsequent to that actually drive the cancer, we don't fully understand. And really, to design a drug to treat a cancer, you have to intimately know the biology of that cancer. We need to know what proteins, what DNA, what RNA, what are the essential drivers that may be upstream or downstream of the GNAQ or 11 mutation that are potent and selective enough that if we turn it off or target it, it'll shut down the entire system. And that's where having this tissue can be really helpful. Now, there's other ways we can do this, like mouse studies and using patient-derived cell lines. However, patient-derived cell lines are not always 100%. They can change and evolve over time. So doing sequencing on a cell line that's been around for 10 years may give you some different information than doing sequencing from a patient biopsy. Those are very different things. And then, in mouse models, sometimes we can probe biology and change stuff here and there, but mice are not humans. And although they're tremendously helpful to helping us understand biology and moving cancer science forward, they also have their limitations. And so the best way to design new cancer drugs for patients is to understand the biology of patients. And to do that, we need samples from patients.
Yeah, that makes sense. And I know like as a patient, some of the things that people in the community, especially when you are in in the metastatic world, some of those big questions that come around is like, well, like I'm say like a supposed to have been a lower risk patient, right? Like, and I'm not speaking of myself. I'm just hypothetically talking about some patients who were told, oh yeah, your biopsy from your eye looked like you were going to be lower risk, and then boom, 7, 8 years later, they're metastatic. And it's like, whoa, like I wasn't expecting that. So hopefully some of the research that comes around and that we can kind of gather all these samples in the biobank, hopefully we're able to understand better why does that happen. Um,
Exactly. And that's some of the things, we've already started to see that a little bit, but there's still a long way to go, right? We know
yeah.
SF3B1 mutations typically are lower risk, but they also make you higher risk for having late relapses, 7, 8 years, if not 14 years down the road, like you mentioned. And so
Mm,
initially, when we understood uveal melanoma, we thought it was just low risk, high risk. Now we know it's low risk, high risk with or without PRAME. We know low risk with SF3B1 is different than not. And so it's using these tissues to develop more information to better refine all the tools and treatments that we have.
yeah, absolutely. I mean, just like hypothetically, like where would you love to see the research from the biobank kind of influence like the development of clinical trials or like the use of treatments in the patient, like kind of journey of metastatic disease specifically.
Yeah, I think there's many ways we could utilize a biobank. One of the things we're seeing in oncology right now is an explosion of drug antibody conjugates and T cell engagers. And what these things are is they're molecules that bind to a protein on the surface of a cancer cell. And on the back, so one part of the molecule binds the cancer cell, the other part has a payload, which is usually a chemotherapy or sometimes an immune therapy or sometimes a targeted therapy. So it's a way of selectively delivering cancer therapy straight to tumors. But you have to know what protein is expressed on that tumor. And we know this very well in stuff like breast cancer, where we see HER2 activation all the time. Um, and there's a lot of these coming out. And so one thing we could do with the biobank is if we collect enough tissue, we could actually look to see, do uveal melanomas express any of these targets that these drug antibody conjugates are targeting right now? And can we repurpose and pull some of those drugs from development into other cancers into uveal melanoma as well? The other thing we can do is really just try to find new targets ourselves. The technology for understanding biology is exploding right now. It used to be that we could test for, we could sequence DNA, then we could sequence RNA, then we could sequence whole exome and whole genome DNA. And now we can sequence, link RNA and microRNA and proteomics and phosphoproteomics, and all of these things, could tell us the story. And putting them all together may be more important because we always like to think that if you have a mutation in DNA, That cut is transcribed into RNA, which is transcribed into a protein. But really, it's more complex than that. You can get DNA going to RNA, but something could knock it down and prevent that RNA from becoming a protein. You could make it a protein, but then you could get degradation of that protein or phosphorylation or some other modification that shuts it down. And so, really, using samples to kind of look at the complete biology from start to finish is what's most likely to give us the insight of how to treat this disease. Now, that's very hard to do. You need very good biostatisticians, very good data scientists, and very good sequencing and multiplexing technologies, but those are coming. We're starting to see those enter the clinic and be more and more useful.
Aura Biosciences is a clinical stage biotechnology company focused on developing precision therapies for solid tumors that aim to preserve organ function. Our lead candidate, Belsar, is currently in late stage development for primary choroidal melanoma and in early stage development in other ocular oncology indications and bladder cancer. Our mission is to grow as an innovative global oncology company that positively transforms the lives of patients. For more information, please visit us at aurabiosciences.com.
Yeah, I mean, it all sounds like there's so many pieces of the science that I definitely don't think that I'm like fully grasping. But I love what you said about how the idea is to help tell the story, right? We're trying to fill in the gaps.
Exactly.
Okay, so um, I want to chat a little bit about like we have at Acure Insight, if you guys have been around, we have a couple of registries, or I guess we have a registry, and then we have the biobank. And I wanna see if we can kind of differentiate between the two and just kind of explain why both are important. Because we do have the Insight registry, but this is the Insight Biobank. So why do you think, like as a clinician, as a researcher yourself, why do you think that it's important to have both of these? We've got the natural history registry plus a biobank.
Yeah, it's important because they help us answer different questions, and the biggest power comes from linking the two. So having the registry, that allows us to answer questions like, is there clustering in families of uveal melanoma? And if that's the case, is it genetic? Is it environmental? It helps to tell us, or it helps us to identify, is there a subgroup of patients who are low risk, like you mentioned, but still end up getting metastatic disease, or vice versa, people who are high risk and those who don't. And that really, so the registry helps us answer those type of questions on a population patient level. The biobank really helps us answer the questions on the molecular biology level. Now, if we compare the two and say, learn about the biology of someone's tumor and then link it to the registry and say, this person was actually one of the high risk people that never actually developed metastatic disease, we can then try looking and saying what in their biology is unique compared to those who did, and that can provide valuable insights into how can we potentially try manipulating other people's tumors to turn them from high risk of metastasis to low risk metastasis. So they're both very important because they answer both questions, or they answer separate questions, but they're merging them together brings us the most power and ability to really try to make an impact in treating uveal melanoma.
Wow. No, that's like, I mean, I think that is kind of just like the most crazy part of the research world that I think exists is this idea that we're trying to piece together all the puzzle and not just like using like one piece of it. Like we gotta pull in the natural history. We gotta talk about what is the patient actually experiencing, even though, maybe statistically they were supposed to behave a certain way. So as we're like looking into the future, like the biobank has only been around for a short time. Like what do you feel like success looks like for the Insight Biobank 5, 10 years from now?
Yeah, you're right. It's really been around just a short time. I think we really only started collecting a couple months ago. And I think, success is going to come in waves. Obviously, to answer the scientific or to start asking the scientific questions, we need the tissue to look at. So step one is really getting the messaging out about the biobanks and hope that patients, are willing to talk to their surgeon, their interventional radiologist who's doing the biopsy, wherever it is in the US, and say, hey, I would like to donate my samples. It's a very easy process. The biobank will ship samples to them. They just simply ship it back. It's very easy. But really, I think the near-term success is getting the message out and starting to get samples collected. And then it's on us as the biobank to start engaging with researchers and letting them know This is the resource that the patients have developed and contributed, and this is open in public. So how can we answer the best questions? And then getting the researchers to know this is available so they can start designing those questions and then start doing our first studies. The other thing that we're trying to do is partner with researchers who've already discovered something and maybe have an idea but need more samples. So one of our initial collaborations was working with Dr. Hawk, who needed some extra samples, and so we put out a blast saying, hey, do you have tissue that can do this? Or are you someone who, progressed on this drug, and do you have samples that you're willing to share? And I think that can be really powerful too, because that just speeds it up. If a researcher has an idea or a hit on something that they think could be used to treat uveal melanoma, they can either collect the samples themselves, which could take a couple of years, but if we can partner with them and speed it up, that's just going to get new treatments, new science to patients quicker. And that's really our goal, getting stuff to patients quicker.
So I think one of the challenging things that patients face, especially when we're diagnosed, is realizing just how rare ocular melanoma is. And then, even in the metastatic world, like seeing how challenging it can be to treat. That can be really discouraging. And I think that a lot of patients sometimes wonder, like, can I actually make a difference, right? I'm just one person. Could my tissue really make a difference? So I love like hearing what you said about how if a researcher has a specific question that they can get answered, like we can, like the biobank can kind of put out a call, right? And say like, we need tissue for this reason. And then I guess as a patient, like that just makes me feel like, okay, like it's not just going to sit in the biobank, like it has a direction, right? It has a specific direction of what it's going to do and who's going to use it and why. And I think that just knowing that the whole purpose of the biobank is to drive, research in that kind of a way, like actionable research, things that are going to move the needle forward. That makes a really big difference for like, why would I want to donate my sample? Because I think that some of it probably comes down to like a lack of understanding of how this works, and maybe even like you said, being nervous to like ask the doctor who's doing the biopsy, hey, I'd like an extra sample. So I guess for those people who are like, I'm not really sure how to go about this process, can we walk them through, like, say let's use me as an example. Like say I've got a sample that I think that we're going to biopsy, and I want to make sure that some of that tissue ends up in the biobank, not just say in like the hospital stores. What's the process like for me?
Yeah, so if a patient's interested, they can go to the INSIGHT Biobank website and they can put in their information and request it. Then what happens is there's a consent where virtually someone from the biobank will walk you through and have you sign an electronic consent about storing the tissue. And that's a great time if you have any questions like, can the tissue be used for? Can it come back to me? Can they know it's me? All of those questions and any other concerns you have, that's the perfect time to answer that. And that consent visit, go to that consent with your questions. You can always say, actually, I don't want to participate, but the only way to get those questions answered is to go to the consent visit, ask the questions, and get it done. Once you sign the consent form, then you can either talk to your surgeon or the interventional radiologist yourselves. Otherwise, the biobank team can try reaching out to their clinic, and then they'll either send all the necessary, sample collection tools, vials to the clinic or to you. And then all you need to do is make sure when you go in for your surgery or biopsy that the team performing the biopsy has that tissue. They put the tissue in there. It comes in with a FedEx label. It's shipped back. Usually the institution ships it. Maybe they ask you to ship it, but you drop it off. It's a prepaid shipping label. It goes back to the lab and it's all done. And so it is very easy. A lot of times in the consent, the thing that we ask our interventionalists to do is to collect as much tissue as they feel is safe without increasing the risk. So a lot of times when we do a biopsy, we do 2 or 3 cores, sometimes 4. Sometimes when you do a biopsy, the lesion's large enough and it's in an area where you could safely do 8 cores. They just don't always need that. But what we tell people is we ideally would like this much, or up to whatever you feel doesn't increase the risk of the biopsy for the patient, because we never want to increase the risk, and we let them determine that, and then we take whatever they feel is safe.
So I know this is going to come up. We have tons of people who, live in the world of like cancer, and we are trying to understand like, okay, does having a biopsy somehow increase my risk for spread? And I feel like you and I have had this conversation specifically a number of times, but can you just kind of clarify again, like that myth around like kind of the, what is it? It's like that if you have a needle point biopsy, that the path of the needle pulling out is going to like somehow be a path of spread. Can you help clarify what we know about uveal's behavior?
Castle Biosciences is a proud sponsor of this I Believe podcast. Castle's innovative uveal melanoma tests provide valuable information about the risk of your eye cancer spreading to help you and your healthcare team individualize the management of your disease. To learn more, visit castlebiosciences.com.
Yeah, so there is a couple of cancers. It's very rare where we know that if you biopsy something, it can grow along the needle track. That is not uveal melanoma. And actually, all of the data we have from uveal melanoma suggests that for patients who will develop metastatic disease, that disease has probably already spread and is remaining dormant in the body even before we find the tumor in the eye. And that's why treating the tumor in the eye may or may not affect outcomes of metastasis. And so, if the cancer spreads, it's probably already done so. It's probably just doing so in a single cell level, hanging out and hiding. To see something on a PET scan, typically you need a billion cells in one spot. So it takes a while for the cancer to grow that much. So doing a biopsy absolutely does not increase the risk of cancer spread or growth or anything like that in patients with uveal melanoma.
Okay, so then when you say like get the amount of biopsy tissue that's safe to do, like is that more like from a physical comfort level of the patient of just making sure it's not too invasive of a biopsy? I guess I'm just trying to make sure that I understand and help clarify for
Yeah.
those of our people listening.
Yeah, exactly. So it's a thing for the patient or also the proceduralist. So sometimes, there's a lesion that's in a tight spot. They may only be able to get one cord. Then we don't collect for the biobank. If they feel like it's going to be risky for just one, then we don't take more. But sometimes it's, some people just have a spot on the skin, and you could either just take a part of the thing or take it all off, and it's really no different. And that's an example where we may get more tissue because it is safe just to take, a larger lesion of the skin versus just a smaller component of that.
Okay, so I want to shift a little bit away from the metastatic side of things, because obviously we've got metastatic disease biopsies that can happen for various different reasons, whether it's to confirm metastatic diagnosis or to join a clinical trial. I know is another setting where you might need a fresh biopsy, like within a certain amount of time. So like, various different opportunities throughout the metastatic journey to obtain biopsies and to kind of volunteer that tissue to go into the INSIGHT Biobank. But if we go back to the beginning of initial diagnosis, how do you like see this working from like an ocular oncology and like the original primary diagnosis point? Because we've got needle point biopsies for like some tumors, and then we have like enucleation, obviously, for others. Proton beam therapy, which is a totally different treatment for other parts of the eye. So yeah, how can we kind of see this working with the primary tumor?
Yeah, there's a couple of things we could do with the primary tumor, but the primary tumor is a little more challenging. So right now we're starting with metastatic, and we'll roll in the primary tumors in the future with the biobank. With the primary tumor, a lot of times if you're getting, proton or plaque brachytherapy, we just do a fine needle biopsy, which really only gets you a couple of cells. To do kind of deep sequencing of DNA, RNA, or protein, usually the more you put in, the more accurate it is. So it's hard to ask broad questions on targeted biopsies such as that, which is why the Castle test is, an RNA test that only looks at, I think, up to 15 genes, if that, because with DNA and RNA, you can ask, if you are only looking at a couple of things, you don't need much tissue. The more things you look at, the more you do. So with a biopsy, you can ask very specific questions, but not broad questions. Now, there is emerging data that vitreal fluid biopsy, the fluid within the eye, which is safe to take, may contain some of the same DNA, protein, and cells as a uveal melanoma biopsy. And that's a situation where we may be able to get more. And there's, I think UCSF and Stanford are doing a lot of research in this space. And so in the future, the biobank may expand into that, since we may be able to get fluid where we could be more useful on a kind of bigger question answer. And then for enucleation, you would think if you remove the tubule, it would be very easy to send it to the biobank. The problem is these are still pretty small, right? Even a large tumor is 2 centimeters. And when we do a surgery like enucleation, where margins are very important, we need to make sure we got all the margins. That's not in the nerve. It's hard to take a portion out of the enucleation to send it to pathology, especially if we think that could affect their determination of did they get it all or not. Because again, the number one thing is we don't interfere with patient care, and so we can't ask that question. We can't prevent pathology from asking that question. So that is a little more nuanced, but some patients you can get that.
No, I'm glad that we talked about that because I definitely believe that this will be a question, especially, as we're able to move to any level of the biobank taking tissue from a primary tumor, like that's definitely going to come up. Because like in my head, I'm thinking like, I've seen the picture of my eye from when my doctor enucleated it. She showed it to me, it looks like a little pomegranate. My head says, well, can't I just donate that eye to science? But I also know that it was sliced through like for multiple different slides because they had to verify that it didn't, extend beyond the actual eyeball globe. And, kind of that was a huge influencing, I guess, bit of the biopsy for my actual treatment plan. So like you said, like so important that it doesn't interfere with the treatment of the patient. I have like one more question, and I guess this is kind of more around like the clinical trial space. If someone's getting a biopsy for a clinical trial, can they also, like, as long as it's safe to do so, ask for additional samples to be sent into the biobank, like for a biopsy of a metastatic tumor?
Yeah, you absolutely can still ask. It comes down to what is safe to collect. So if they're, sometimes when we do a biopsy for research, we need a biopsy for pathology as well because we're sending, circulating tumor DNA and we need tissue for that. Then we need to collect tissue for the study, and then the biobank would come third because we never interfere with the patient care in the first two or more patient care. And so it might be that for smaller lesions, they can't get enough safely. It could be that it's a large lesion and they absolutely can, but it's absolutely a question that can be asked. Yep.
Okay, awesome. Well, I feel like this has been super enlightening. What do you hope that ocular melanoma patients today understand about the future because of a resource like the INSIGHT Biobank?
Yeah, the goal of a biobank like this is to really help investigators find better ways to treat the cancer in the future. And so we're really hoping that, we can use this to develop new therapies for future patients. I think the thing that, well, I think one of the unique things about the uveal melanoma patient population, which is one of the reasons I love working with this group in the INSIGHT, is just how much drive and support there are from the patients to partner and cure this disease. And I think that's super unique. And I also think it's incredibly important. When it comes to rare cancer, no one person's going to cure this disease. No matter how great that one person is, it's going to take a team. And I'm not aware of any other disease where the patients and the advocacy groups are such strong partners or members of the team as uveal melanoma. So I think a lot of it is just keep doing what you guys are doing because you guys are already leading the field.
Well, thank you so much, Dr. Moser. We can't thank you enough for just all of the continued research that you're doing and for the treatment of the patients that you have on the west side of the United States. And we're just really grateful that you're willing to be a part of the team with all of us. And I love what you said about that. And just to kind of close out, I want to also make sure that we thank our sponsors. Thanks to our listeners. And if you haven't yet, make sure that you subscribe, hit that little button on YouTube or on your podcast platform, and share this with some friends. Like Dr. Moser said, the best way that we can get the biobank to move forward is to have more samples. So the more people who know about it, the better. We'll also put the link to the biobank in the show notes so that you guys have access to that and you can share that with friends. And, let's just spread the word because that's how we're going to help the needle move forward on research. So thanks again, Dr. Moser.
Thanks for having me.